This is my first opportunity to thank the organizers for the invitation, and I think I really appreciate being here. And let's see to try to do something with lights. Too much? Is it okay? And as we discussed with Abba what we could do, I decided that given the overall talks that we're presenting, I was going to talk about models for schistosomiasis, although I'm not an expert on that. So I never worked really on schistosomiasis. I did something on models for parasitic worms, but nothing really for that. So I'm sure somebody in the room knows much more than myself about that. This is really meant as an introduction for people who don't have experience on that. And okay, there are, as you all know, there are many diseases that are caused by parasitic worms. So here I listed some that are the first ones that are listed in the list of the WHO about neglected tropical diseases, and actually most of them are concentrated in Africa. So most people who suffer of from these diseases are concentrated in Africa. In particular, in about schistosomiasis, a review of 2002 showed that about there were 200,000 deaths each year due to schistosomiasis. So it's something that is quite relevant for human health. And even if that's a few years ago, there are the number of people that are infected with that is probably in 100 millions, more or less. It is a disease that it's curable, so there are drugs that work, but it's still it is very widespread because it's difficult to be eliminated. And this is just a picture that I found on the WHO on the WHO site about the prevalence of schistosomiasis. So it's concentrated in particularly in a few countries in Western and South Africa. And actually there are several species of schistosomiasis. There are two main forms, one that is called the intestinal and one that is called the urogenital. And most models that I've seen are concentrated on Schistosoma mansoni, that is the intestinal form that is widespread in Africa and somehow in South America. And there are other species. Okay, so what is particular about schistosomiasis relative to other infectious diseases that we are discussing? Well, this is a sort of a life cycle, and I saw that exactly the same slide was shown by Corey Morin. And this shows more or less for those of you who are not familiar, how things work. So if we start here with the eggs that are being laid and arrive into water, then the eggs hatch into some other structure that is called miracidia. The miracidia penetrate into snails when they are in water. If they find a snail, they penetrate in the snail. They go into some transformation and replicate quite a few number of times inside the snail. So from a single one, you get a replication. Then they release some other larval form that is called cercaria. They are again in water. And if they get if they find a human, they penetrate into the skin. And after a certain time, they become adults. So here are still larvae. They become adults, and when adults, they find a place in the intestinal for the one that has to do for the intestinal form, or near the bladder. In here, they mate because they are a sexual form, and they go on releasing eggs, and they can live in the intestine for several years. So this is more or less a cycle. From the point of view of modeling, as you see, something that you get when you in water. So for exposure to water, this is clearly, as Corey was saying, related to water. What are the important things from the point of view of modeling? Well, one important thing is that there is an intermediate host. It's in some sense, it's from the modeling point of view, it may not be so different from a vector, but still it's something that one has to take into account for modeling. A second thing that the worms do not multiply within a human. So there are larvae that transform into adults, they mate, but then to have multiplication, they have to go through the cycle. And also an important thing that the morbidity depends on parasite burden. So it's really something relevant only when the parasite burden is high. So you want, there are two things to consider because of this. One is that you have to consider not only whether an individual is infected or not, but you want to consider the level of infection. So so this is one of the things. The other point is that you need to consider reinfections. While in the other epidemic models that you have seen, there are infected people and then it's not important whether they are re-exposed. In this is essential because this is what has the level of parasite level to grow. So these are the two main things from the modeling point, well, intermediate host and this. There is something else that most models neglect also because it's, as far as I understand, it's difficult to quantify, is the role of immunity. Certainly there is exposure to worms cause a level of cause an immune response. This depends also on age, and this should somehow consider. Very few models really consider that also because, as I understand, it's not easy to study experimentally. So this is more or less the structure. So what is one possibility of modeling this? One possibility is to use as variable the density of hosts, in this case humans that have I hosts. I'm trying to find this one. Oh. So in principle, one could have as variable pi of t. Pi of t will be hosts with I parasites. And then you can have a host that goes from I to I plus 1 by acquiring worms. And probably you could say that this is proportional to the density of Miracidia. So you can go from, you can acquire a new worm going from state I to a state I plus 1 by acquiring Miracidia. And then you can go from I plus 1 to I because of the death of the parasite. So this is the main structure. And then you should add something related to the death of the individual that will be depend in most on, there will be a natural rate that we call mu h, the death of humans, plus there might be a death related to the disease that is often treated like that. So these are simplified assumptions that tell you what is more or less the process. From these assumptions, you can arrive at writing a differential equation that I'm not writing in the whole form, but more or less looks something like, if we look at how the density of individuals with I parasite change, well, there is a gain from the one, there is a gain from the ones that have I minus 1, because there are the individuals that had I minus 1 and gain 1 individual. There is a gain from the individuals that had I+1 and lose 1 because it's lost, and then you have a loss term given by all these factors. So death of the parasite, new infection, and something else, and the death. So one can write, just take into account the number of parasites an individual has, a system like that. Of course, this neglects a lot of important things like immunity, susceptibility to infection, perhaps other heterogeneity in the population. But one could write something like that, and as it's written here, it would be quite complex. Would be quite a complex system because you would have a very easily a very large system of equation. In principle, could be an infinite system. There has been some mathematics about that, but certainly they are not the easiest model that one can think. So what has been done? One thing has to look empirically at what is the distribution that one sees in the population. So here, for instance, are some data that were collected, I think around 2000 in Brazil. What does this picture show? Well, that in the population there was a large number of individuals that had no detectable parasite load. Doesn't mean that they didn't have no parasite, but at least no one could detect it in the feces. And then there was an increasing number of individuals that had a very high parasite load, at least as measured by eggs per grams in the feces. So you have a distribution of this kind that is that says that the parasites were very aggregated in a few individuals. There were few individuals that carried most of the parasites. So this is generally a property of this. And what something that has been typically done is to fit a distribution to this, in particular a negative binomial. So it's really a bad picture. It's not my fault, but it's the distribution. There are some dots here that it's very hard to see them even printed, but there are some dots of distribution that actually fit this distribution quite well. This mass has been obtained by putting together all individuals whose load was beyond a certain level for statistical reason. And you have this distribution has some property that we can look at them statistically. One of them is the average parasite load that, because of how it was measured in units in 12, so it was measured as, say, more or less 100. And then there is this other quantity that is called kappa, that is a parameter of the negative binomial, that was estimated at this number, 0.2. What does this number mean? Well, I know several of you know well these things. The negative binomial is a probability distribution, has a certain formula that one can find. The easiest way, the shortest way to remember is to remember through the generating function. And from the generating function, one can find everything. This distribution can be parameterized in different ways. One of them is to use the mean and this parameter kappa. And this is the formula. This doesn't tell you very much. The only important thing to understand is when k goes to infinity, this is a classical limit, and you get the Poisson distribution. So if kappa is very close to infinity, it is a distribution that is not very aggregated. It is a prototypical random distribution, and so there is and it becomes a Poisson distribution. The closer is kappa to 0, the more the distribution is concentrated in a few individuals. So 0.24 is quite close to 0. So this is an example of a distribution concentrated in a few individuals. The idea of many models is to say, okay, we know we have this distribution, and that seems to fit the data, and we assume that this is a distribution of that the distribution is always negative binomial, and sometimes we also assume, well, many times, that this number kappa is a property of the population and the thing, and cannot vary, something that is fixed. It's clear that it's a simplified assumption, and that people have also tried to do different things, but it's one of the classical ways of doing that works. Of course, the fact as it's written here, the distribution fits the data doesn't mean that it really is a good model for what happens. So having made this assumption, one can use some statistics to describe what is the population. And the simpler way to describe the population is to describe it through total population density, that is called N, and total parasite load. Instead of P, one could use P divided by N, so the average parasite load, that may be something easier to feel, but basically they're identical. If you assume that the distribution is a negative binomial with a fixed kappa, and you go from this equation, so you write this equation and you do some algebra, and we don't have time to do that now, but if you go through that, you arrive to a much simpler system than you think. So it becomes a system in which you write, okay, the population will have an input that here is written as a fixed input. One could decide also something different, but let's decide there is a fixed birth rate, and this model assumes that all new individuals are born without parasites. There is a death rate that we say mu h, and the death rate due to the parasite load that we assume that was proportional to parasite load becomes simpler terms like that. The equations for the total parasite load is there is an input that comes from encounters between individuals and Cercaria. C of t stands for Cercaria. The death rate depends on the death rate of the hosts, the death rate of the parasites, and the death due to parasitism. And there is this extra term that this is where really the negative binomial assumption comes in, that is hard to understand, just to justify, but it is the extra decrease in parasite load due to the fact that individuals with high burden will tend to die. So this is, and of course, here is the only point where the parameter kappa enters. And just to remind you, C of t is the density of Cercaria, alpha is the disease-induced mortality, and well, kappa, you know what it is. So this is the structure of the system, but of course this is not a closed system because there is a C of t, the Cercaria. So we need to say something about how the Cercaria does. Clearly, if you want to apply this to a human population, probably you shouldn't neglect age. So you should include age to make a more realistic model. But this is just to have a more tractable mathematics. So we need to look at the other part of the cycles, because Cercaria will be produced by infected snails, and infected snails will be produced by eggs that are hatched from that are laid by infected humans. So you need to look at the other part of the cycle. This part, in some sense, is simpler because since the sporocytes multiply within a snail, it seems quite reasonable that we simply want to distinguish snails between susceptible and infected, like in classical model. We don't need really to distinguish their parasite load. That doesn't seem to have a strong influence. Perhaps one might want to have an intermediate stage. Here it's not written, because generally it takes some time for the for the larvae to grow in the snails and to be able to release cercaria. So it might be useful. It might take a few weeks, so it might be useful to introduce another term. Here it's not written, but it's something. It has been known experimentally that infected snails suffer both from increased mortality and from partial sterility, so one should include that. And to go closer to the themes of this workshop, snail population dynamics is certainly influenced by temperature and water level. That is something that is going to be affected by climate change. And I'm not really talking about that also because there is very little that I know has been done on that, but this is something that is definitely worth studying. So how would the equation look like? Well, the equation for the susceptible will have a birth rate, a death rate, and the susceptible will be infected by encountering Miracidia. So it is more or less a classical term. And infected individuals will be created when susceptible is infected, and then they will die with some mortality plus some extra mortality induced by this small letter t seems to mean in principle this should depend on time through temperature and climate, but it's small, so I'm going to ignore that. But it's this is something that we should look at. What has been, so we have a system without having the dependence on time that is like that, and still there are two parts that have to be completed. What is this function B, the fertility function, and the Miracidia? In models, there are two different kinds of assumptions that have been made. One is to say, okay, snails have a constant fertility. It doesn't depend on anything, it's just a constant, and this, of course, simplifies the model very much. Another possible assumption is to say there is partial sterility, and in this model it is assumed that it's total, so infected individuals do not give birth. Only susceptible ones give rise to birth. And then there is intraspecific competition, so birth rate decreases with population size. This is one possible form that looks like the Beverton-Holt choice that was suggested by Jacek, but there are other possibilities. And of course, one could think if one has good data, one can try to find a better form that describes better what's happening. How about Miracidia? Well, Miracidia, in principle, could also have a differential equations, because Miracidia are developed from eggs that are laid by infected humans. So there is a production term that in first approximation can be assumed to be proportional to parasite load. Miracidia will die in the water at some rate, and then they will be and we will encounter snails and will infect them. So one could have an equations like that. However, this is generally thought that Miracidia live a very short time in water compared to all other terms that we are discussing, so one uses what is called a quasi-equilibrium approximation, so say, okay, this equation will be more or less at equilibrium, and so we have something like that, just by putting this equals 0. And actually, generally, there is even a stronger assumption that is made. The assumption comes from the assuming, well, this term delta m is much larger than the other one. It is much more likely for a Miracidia to die than to encounter a snail. And so we say, okay, we can neglect this term, and then mt is simply proportional to the parasite load. So this assumption assumes that the density of Miracidia in water is proportional to the total load of a parasite in the human population. And this is clearly simplified the system. And similarly, one can assume that the density of cercaria is simply proportional to the density of infected snails. And these are simplifying assumptions. So the system now will be complete because we have the equations for x and y that depend on the equations for p, and the term c that was needed in the equation for human comes from that. So we have made a complete system. Just two cautionary comments that, as I discussed, this assumption that density of Cercaria is proportional to current infected density of infected snail density is a great simplification because neglects delays and neglects the fact that Cercaria production is actually not constant in time if you see that experimentally, so it might not be a great assumption. And also something that all people that work with parasites tell, that actually egg production is not proportional to individual parasite burden because there is a lot of, there is sexuality, so you need at least 2 worms to start. But also there is an effect of competition in parasites and the immune response. So actually nonlinear form must be more closer to reality. So now we have the complete system. This one I've written comes from a paper by Jilan Feng, Fabio Milner, and Lee of 2002. This would be, well, it's actually slightly simpler than their system. So this would be the system that takes together all we discussed here, and is a system of 4 differential equations. So it's quite simple relative to the complexity of the system. And as you know, there is reproductive ratio. Reproduction ratio is one of the key things in epidemic models. So one can count and what I call an RMC, so the average number of Cercaria produced by a newly produced Miracidia, and this comes from the probability of being able to infect a snail. Okay, I mixed them up, but it's not. So this comes. It's the opposite. It's if from human to snail. So the average life of a new adult parasite, the average number of Cercaria produced. Well, you can adjust them. So I mixed up the terms, but basically we're counting how many you should change the denominator. Basically, you say how many the number that it goes from Miracidia to Cercaria and to Cercaria to Miracidia. And the threshold quantity is quite easy. You want to have that the total number is greater than 1. So if you put both of them together, if you look the number from, you go from human to snail and then from snail to human, the total number has to be larger than 1. And the system is not completely easy, but there are some of the properties are standard epidemic models. So if this quantity, the product of reproduction ratios, is less than 1, the parasite extinction is certain. Instead, when this quantity is larger than 1, there is an endemic equilibrium that is unique. So it's, even if it's 4 differential equations, it's not terrible. If you assume that B has a more complicated form, where we assume that there is partial sterility and competition, the endemic equilibrium is not always stable, and the system can easily give rise for reasonable parameter values to Hopf bifurcation and periodic solution. And if you are more interested in the mathematics, you can try and look at what has been done. without going into more discussion on this, we can look at a simplified model. The simplified model was actually proposed several years before by Woolhouse, and is based on two main assumptions: that we neglect death due to parasitism, so we set alpha equals 0 and the human population size is constant. One comment, it's true that compared to other sources of death, this is quite small, so from the point of view of demography, one could probably neglect it. However, it's still important in the dynamics of the parasite, at least in this model, because it influences the distribution of parasites. So the fact that there is a disease-induced death is something that limits the number of parasites that can grow in the model. Then one can think there are other important terms. And the other assumption is that snail population is constant. And so this is... okay, now the first equation does no longer exist, so the equations for n. It's easier then to write equations for variables that have a better interpretation. So the variables become the average parasite load and the proportion of infected snails. And snail population is constant. This is a natural variable. I just look at the proportion that is variable. And if you take the equations that were written before and you put these assumptions, then the equations simplify quite a bit, because you simply get an equations for parasite load. You lose the complicated term with alpha and kappa, so you simply have an increase due to encounter with infected snails. So y of t is a proportion infected, and S is the density of snail. And then you have a death rate due to parasitism. And as for infected snails arise by susceptible snails, 1 minus y of t, encountering parasites. And parasites, from all these assumptions, we know that miracidia are proportional to density of parasites in humans, and this is bar n x of t. So the system in these assumptions becomes extremely simple. It becomes extremely simple, and what I said before about about endemic equilibrium, you can compute it explicitly. So from this number that we discussed before, we have, okay, then the mean parasite load at equilibrium is simply the quantity RMC minus 1 over RCM. And if the product is larger than 1, then parasite load is positive as we want. And the density of infected snails is simply 1 minus 1 over the product of 2. So 1 minus R0. And this system is very easy. You can show that this is always globally asymptotically stable. So you can say everything. And this is, these equations may be interesting if you want to understand what happens with therapy. And suppose there may be other ones, but there are two main ways of, well, I said the modeling therapy, but it's more modeling control. So one way of control is through therapy, because if we give drugs to people, this will kill worms, and beyond improving their health, will also decrease the load on the burden of worms in the water. So there is this effect. And so now there is a campaign for mass drug administration. And we can say that we can model this very simply by we increase this kind of therapy. Simply means that we increase mu p. Perhaps if this is done not continuously, but only a certain amount of time, there are campaigns, you could do that periodically. But the idea that mass drug administration can be modeled through an increase of parasite mortality. The other kind of intervention that could be is to have to try to decrease the snail density by putting molluscicide in the opportune body of water. The effect of this would be possibly to decrease snail density and more certainly to increase snail mortality. And what would be the predicted effect? Well, if you remember what are the relations, so basically mu p enters in what I called RMC, so it enters here. Bar S enters in the quantity here, while mu S enters here. So one can get some sort of idea. If we are able to decrease the snail density, decreasing snail density will have a linear effect of decreasing RMC, and this will decrease linearly the prevalence, the mean parasite load. The effect of mu p will be more or less similar because since in general mu p is much larger than these two other numbers, if we increase mu p of a certain fraction, we're more or less decreasing RMC of the same factor. It's not exact, but more or less so. So this would increase. On the other hand, the effect of increasing snail mortality by itself is less important because it's true that increasing mu s will decrease RCM, but as long as this doesn't bring the total reproduction ratio below 1, the effect of decreasing this is not so important in the mean prevalence level. So molecular treatment is relevant if it's able to decrease snail density. If it just increases mortality, but snail density is compensated by other factors, this is a less relevant thing. And clearly, these simple relations are true only in this simple model. In this simple model, we can compute everything, and we can say, okay, there are these simple relations. As soon as we go to a slightly more complex model, as the one I discussed before, this doesn't hold anymore. So one can look at how different assumptions will make deviations from this simple fact. So for instance, some of these, in the model by Feng and Milner, some of this quantity depended through the square root in the equilibrium. Another difference between the simple and the more complex model is that if the model assumes some snail dynamics and some compensation, is that it's possible that one might get periodic solutions, so terms of increase and decrease that are not related to therapy or other, which is something that cannot have in a simple model. Something else that okay, in principle, the parasite load will depend on the aggregation if you have a more complicated model, if you take this into account. And also something that you might be more interested than in mean parasite load, you might want to know what is the proportion of the population whose level of, whose burden is beyond a certain threshold, because this might be more important from the health point of view, and for this you need some information about the distribution. Moreover, if instead of deciding that parasite aggregation is some constant that has been fixed, but you try to understand which are the processes like heterogeneity, age distribution, or what that cause aggregation, then you might understand better what you expect that would be a response to therapy. Not that kappa is simply constant, but perhaps this has more complicated effect. So this is sort of, I'm not saying anything about it. I saw there are some very recent papers that have a much more data-based approach to to modeling schistosomiasis that are quite interesting. I don't have neither the preparation nor the time to discuss them, but more or less they discuss more in detail several things. So for instance, there is a recent model by Gurari and Dole in which instead of using this simplified relation, they have something of this kind, but not completely, in which they discuss, okay, we divide parasite burden in certain strata. So we group parasite burden. we assume that there is a nonlinear relation between parasite burden and what we can actually measure. So we can actually measure egg in feces, so what is released. And from these relations have been calibrated from data. And then you can look at short and long-term simulations, for instance, of mass drug administration control strategy that take into account also periodicity and other things. So this is something that if you're interested in schistosomiasis, you should certainly read. There is another paper that has also, well, this is the paper by Gurari, somewhat more complicated, but more or less in the spirit of what I talked. This other paper of Mitchell and Tal instead is quite different instead, because it is based on individual-based simulations. So each human is simulated and has a certain age, a certain parasite load, a certain immune response. And the model has been calibrated looking at antibody response before and after a treatment in Zimbabwe, so it should be somewhat calibrated to that. One thing that is in this model is totally absent, that this is only a model for humans, and so there is no coupling to snails, and so it is assumed that Cercaria density is always constant, independent of what we do to humans, so it's not really studied what will be the effect of control in general, but simply what we can expect as effect of a strategy on immune response. Starting from the point of view that part of the infection is controlled by immune response, is trying to look at this point. And another recent paper instead has a different approach. So it basically uses the model I showed or some small modification of that, except that it is a patch model and the number of patches is very large. It is based on the actual map of Burkina Faso. And beyond the patches are connected by human mobility. So humans can move from one patch to another, transporting the worms they carry if they do. And there is a hydrologic transport of larvae. So larvae may be carried by water from one patch to another. And so it is a rather complex model from this point of view that tries to produce a risk map of the area. And however, one important thing is that all these models actually neglect snail population dynamics. It generally is taken as a constant in this model. So even if there is, in the Gurari model, in these models, there is a coupling between infection in humans and in snails, snail density is simply taken as something that is given and is constant. Whether this is relevant, I found a very recent paper that tried to measure snail population dynamics actually over a short time period in several sites in Burkina Faso and to fit model to that. So these, I think, are different water bodies, and I think these are two different snail species, one that is the intermediate host for the intestinal, and the other one that is the intermediate host for the urogenital form. And these are in a perennial stream, and these are some other kind of ephemeral body, and this is an irrigation canal. And you can see that the distribution is quite different. Well, for instance, when there is no water, there are no snails. So this is something that one can expect. And what they have done is to, well, you have the data, and these are models fitted to the data, and it is a rather complex thing. But the idea is that all the models include some kind of density dependence. So snail population dynamics seems to need density dependence to be explained, so something that can produce more complicated dynamics. And there are different environmental covariates that are important for explaining that. And this depends on the fact. So for instance, for perennial streams, it seems that water temperature and conductivity, that is a proxy for nutrient level, are the most important determinants, while in more ephemeral bodies, other things like air temperature may be more relevant in describing the dynamics. So the idea that I found from this is that actually snail ecology is quite important, and it's also very local. So it's very difficult to give general features of that. So this is just my conclusion. I think I left some time for maybe discussion. So what are the variables that we need to describe a parasitic disease? At least we need mean parasite burden, but probably we may need some other information about parasite distribution and what causes that. There are many steps involved, and from very simple models, you get very simple predictions. So maybe they are wrong. I mean, probably, certainly they are wrong in exact form, but still they may give you some useful insight on what you may want to do. If you include more processes, you can refine the prediction. And also, in some sense, they can be easier to parameterize. It may be difficult to say what is the relation between the density of Miracidia in water and the human parasite load. On the other hand, you may try to find what determines that, what is the death survival or things like that, and egg production, something easier to think. And the trend is towards making very large, complex database models, and this is something that cannot be discussed here, but it's something that you should do. And the other point is that snail ecology is probably important in understanding what is the dynamics, but it's also very sensitive to microhabitats. So it's hard to say, I make a model of snails in South Africa or in something else. We should really look at where we are looking at. Or perhaps trying to have a general model with a lot of different habitats and many different forms, and have some transporting within. So thanks for your attention. Well, the last point is that there is some work has been done. I think there is one point in which there is a lot of work that can be done on modeling. There is something has been done, but I think not very much in being really helpful and important with data, and I think I encourage you to think. And I think there are here people that have access to data. As I told, I never studied schistosomiasis, but I talked to some people who are working with that, and so probably they could be interested in sharing data and discussing how to model that. So I think this is quite important. So thanks a lot, and this is just a list of the references if you want to look. Please?
When is a model data-based?
What?
When is a model data-based?
Well, I just use as a catchword. I don't mean as something precise. I just saying that what I mean is simply to say that the model includes a lot of information about based on detailed thing. In some sense, for instance, to give the example of the model on the geographic model, the model is database in the sense that they did not write a matrix, a random matrix of numbers between patches or something that depended on distance. They used data they had on human mobility and on hydrographic flux to try to decide what were the fluxes. But of course, it's database doesn't mean very much. Was that a question? I just mean that is something that tries to take into account a lot of data which the researchers have access. not trying to simplify, to try to find simple simplifications of reality.
So the model by Feng and co-author predicted some oscillations?
Yes, could predict some oscillation.
Yeah, and then this simpler one is a constant equilibrium.
Yes.
And I was wondering how these kinds of predictions relate to time series data on regional or greater scales. Are the outbreaks episodic, or do they actually oscillate, or do you know?
Well, I hope there is somebody here that knows more about that than me. I'm looking around for experts.
Where's John? Oh, yeah. That was a question for you. John.
No,
Honestly, my answer is I don't know.
yeah, okay, so we'll find out.
But I thought the oscillations are only possible if you use a Beaverton plot.
You need to have oscillations. You need the fact that fertility in snails is not just constant, but it depends on population size, and which is quite reasonable assumption. I don't know.
Yeah, this is going to take us to different approaches we need to incorporate to statistics, considering that each species in any part of the world can behave in a different way according to the physical cycles. So this is going to give a huge complexity to any drawing, because each species will behave in a very different way as any human being here. We could look for a comfort index, a temperature, in which each person in this room feels comfortable, and probably if we calculate the average, no one will say this number in the room. So these oscillations are very related to seasonality and biorhythms, and probably at many scales, at a macro scale globally, and also in microbiology. So how to incorporate this into these statistical models? It's a complex, very complex issue. But we have to look at physical models in order to understand what parameters, how much energy a plant needs to start growing, because they need different amounts of energy. How much light we need to change our mood, because we are different, or to suffer the impact according to our immune system. There are people, medicine is looking constantly at people who got diseases, but I consider that if I were a medical doctor, I would like to look at people who never got a disease. Why? They do not get sick or ill. So these new approaches, that's why multidisciplinarity is going to be so important in order to fit the statistical models to the physical reality. That is my point of view as a biometeorologist. And water, salinization, any physical factor can be a key issue in order to understand why an airborne disease is behaving in a particular way. If you don't look at the air, how can you understand how this disease is transmitted? That's my point of view.
Yes, if I can make a comment on this, my final point was to say, well, either we want really to look at a very micro scale, so we are interested in this village, and we know that there is the little stream around that has this property, and we know more or less, and we want to look at that. Otherwise, if we want to look at a more global scale, what would be the effect, we have to take into account that the environment will be very heterogeneous. So we cannot talk, there is a certain number of snails follow a certain equation or something like that, because there will be a statistics of different water bodies with different features, and somehow we should be able to take into account the distribution of what happens in understanding the global thing. That's my feeling from that. And of course, perhaps there are clever ways of doing that instead of having a very big computer that takes everything into account. Perhaps there are clever ways of summarizing that and taking this into account.
Okay, one more time.
So I find you've got it right there at the end. There's this classic dichotomy between approaching something at a microscale, which makes total sense here, because if you don't have a stream or a pond in your village, then you're probably not going to be affected by schistosomiasis. But then most villages will have that. But if you can break the connection between people urinating in the water or close to the water, then you may be able to break that association between the parasites and the intermediate hosts. So it makes total sense to manage it at a micro scale, but then obviously water is connected across much larger scales. So unless you tackle it at a larger scale as well, you're also probably not going to be successful. So there's a problem with that dichotomy, that you have to manage it at both scales to have any impact.
Yeah, well, I think this is quite correct. This is one of the challenges of being able to model something. Well, probably many processes have that thing, but that certainly is a challenge. Well, of course, it's clear that it's mainly a disease of poverty. So if you have a good sewer system and something like that, this would solve the problem. I think we know that. And
well, I speak as somebody who has been infected, and one of the problems that you get is that it changes the behavior of the host. The parasite changes the behavior of the host, and you basically want to pee into water. So when you're around water, or if you're in water, you're actually more likely to pee. So a good sewage system is not necessarily going to change it.
okay.
Maybe you can add an equation.
So I just wanted to check if we can say then we have something that is key, that is missing in these models, like the socio-economic factors. Are they taken into consideration? Because then his point will come into play, that if we are more developed, or if we have water supply, improved water supply and sanitation, that will also disturb the cycle.
Okay, I don't know how to answer, so I see that he wants to say something. I hope he has.
This is the last question.
Okay. I know I can. Yeah, you're right.
This is a very good point, because I could tell you that when we had a heat wave in Europe in 2003, many people died. All people because of this heat wave in France, in Spain. The impact was huge. So we were looking at many different models, trying to find the relative risk of each 1 Celsius degrees in relation to the people. But there were some sociologists that they told us that our social structure was very important, because now many people in big cities are living alone when they are over 70 years old. So, and in the past, they were living with their families. So their families were looking after them. Because these people, they have dysfunctions in the hypothalamus. And this is telling them if they have to drink all of water, so that they don't feel the heat. So if they are in the family, you will see your grandfather is red, or maybe you will tell him you have to drink water. And because of the way we are changing the social system in Europe, the impact was even higher than how it had been with the social structure were like 50 years ago. So social issues are also very important
Okay. Can you